So today I'm going to talk a bit about microscopy, and in particular optical microscopy. So why am I talking about microscopy? Well, it's an important part of biology and one of the founding stones of biological studies. So microscopy allowed us for the first time to look at cells. So the little bits that make up a living being, whether it be a plant or an animal. And there's some quite interesting and in places amusing history around microscopy. So I'm going to go through the history and tell you a bit about how microscopy works and what it allows us to see. And then next time I'm going to talk to you about electron microscopy. So, this is a microscope. It's not a particularly good microscope, but it is nevertheless a microscope. And there's a stage, an objective lens, a condenser lens, and the light goes through the stage and is focused till you see it through the eyepiece. And I'll go through that in a bit more detail in a bit. So, a little bit about the history of microscopy. Well, simple microscopes have been around for a long time. That's individual lenses like a magnifying glass or like a pair of glasses. But compound microscopes, so microscopes similar to this one with more than one lens, are a bit newer. So we're going to be talking about compound microscopes. So this story starts in the 1620s, and a Dutch guy called Cornelius Drebbel who was living in London at the time showed the first compound microscope, and I just love the description of this microscope. So it's described as being 18 inches long, so that's a foot and a half, I don't know, it's quite long. 2 inches diameter, so probably about that size, and supported on 3 brass dolphins. Don't you just love the brass dolphins? Things have got smaller since then. I mean, this is probably, what, 2 or 3 inches long? 3 inches long maybe? Half an inch diameter. And it is not supported on dolphins at all. So, bit of a shame that. I would have liked a microscope with dolphins, but hey, that's not as it is. So, he was the first compound microscope. Actually, that's not true. People had had compound microscopes before. He was the first person to document a compound microscope. So we'll call him the first compound microscope because we've got no better indication. So, the next place we go, or the next time we go, is 1665, and this guy called Robert Hooke. So he was fascinated by microscopy and by things that are smaller than what we can see with our eye, and he basically had, I don't know, a shed, garage, man cave, whatever, and he did a lot of his microscopy outside of the lab. And he published a book called Crew Grafica, which was a load of drawings of what he'd seen under the microscope. And that was that was the first kind of publication of microscopic images, and the camera technology that was around then, if there was any technology, was large, expensive, and had longer exposure times. So he drew what he saw. And he also came up with the word cell to describe what something was made of. And he looked at anything he could find. So he did a lot of studies of his own body. He looked at skin cells, he looked at cheek cells, he looked at his tears, he looked at blood. Basically, if he could get his hands on it, he looked at it under the microscope, because he was seeing the world in just so much more detail to what had ever been published before. And so cells are really small. So if this is an onion, or it's part of an onion, I've cut it up already. And if you gosh, that's separated easily. If you separate the onion, inside is a membrane. Oh, come on. And you can peel that. It's a little bit fiddly, as I am showing. This is a good demonstration of how fiddly it is to dissect an onion. But I'll get there. And the membrane in between the layers of an onion, oh, that's a nice piece there, once it's come out, is 1 cell thick. And this is the membrane. And if you look at that just with your eyes, it kind of looks almost like a continuous sheet, but maybe with lots of little dots on it. And those little dots are the cells that make up the onion. So he looked at, and actually the first things that he categorised were plant cells. So plant cells are a bit different to animal cells, and we'll talk about cell structure in a later video, but they have a very defined structure. And what he saw looked kind of like this. And it was actually plant cells that he was looking at. It was bark, I'm not sure what tree of, but it was a tree bark. And it was like this. And he thought that these looked like the cells that monks would sit in when they were in solitary confinement, or maybe like what we put prisoners in. So he called them cells. And that's how the term cell came to be used, and it's continued to be used today. So that was Robert Hooke, and as I say, the stuff that he drew, that he'd seen under his microscope, it was really quite extensive. So that's 1665. In 1674, there was another Dutch guy. So Cornelius Drebbel was Dutch, living in London, and this was Anton van Leeuwenhoek. So Anton van Leeuwenhoek, and he looked at microbes, so bacteria. And he saw bacteria moving under his microscope, and he called them animalcules. So cule, as in molecule, meaning little, and animal, well, we know where animal comes from, because he thought they looked like little animals. And they were the smallest thing that you could see with a light microscope. And we're going to talk about that in a minute, because that all comes down to resolution, which I'm going to explain to you. And then nothing much happened for nearly 200 years. But then in 1846, Carl Zeiss came along. You may have heard of Carl, or at least of Zeiss. Because he was a lens maker. And for the first time ever, you could buy a microscope over the counter, probably for hideous amounts of money, but you could buy them. And the Zeiss company is still going today, and in fact I'm wearing Zeiss lenses in my glasses now. So that's Carl Zeiss, but you can see here it's nearly 200 years between the first publication of results found using a microscope to the production of a microscope that you can just go and buy. So that's a bit of history about optical microscopy. After this we get into electron microscopy, which I'm going to do another time, but we're going to talk a little bit more about the theory of light microscopy, or optical microscopy, which is a much more accurate term for it. Now, so if we go back to our little microscope here, as I said before, you've got the two lenses, the objective and the condenser, and the condenser is the one that you look through. The objective you can change to get different amounts of magnification. So what we have is our sample on a stage, which this is the stage here, and we've got sample clips, and we can put our sample, this is the one that came with the microscope, under the clips and look at it through the microscope. So here we have an objective lens. And here we have a light source. So let me attempt to draw a light bulb. Did I tell you that I'm not very good at drawing? There we go, that's a light bulb. So we've got a light bulb and the light is going through the sample. So it's blocked at the dense bits and able to pass through the clear bits. And this lens bends the light coming through it so that it's like this. So if we look here, it would be focused. But we're not looking here because we want it more magnified than that. So we have another lens, the condenser lens. And this focuses all these beams of light up to the eyepiece. And we look through the eyepiece and what we see is a magnified version of what's on the stage. So, I want to talk to you a little bit about magnification and resolution as well, because as I say, bacteria were at the limit of the resolution and they were being seen in 1674, which is really quite early. So magnification is the amount bigger something is. So and microscopes are marked with the magnification of them. So this one, the biggest objective lens here, gives a 900 times magnification. If we turn it round, the smallest is 150 times magnification. And that is how big something appears. And quite often on better microscopes, obviously not on that one, you'll have something called a graticule, which is a calibrated grid, and it's on the stage of a microscope, and that tells you the size of what you're looking at. So it might be 1 millimetre, 2 millimetres, 3 millimetres, 4 millimetres, etc. And that's the actual size of the thing you're looking at. However, because it's magnified, it appears bigger than that. So if we're looking at something that's 1 millimetre and we're looking at it in that microscope in the 900 times objective, what we're seeing is 900 times 1 millimetre, so that's 900 millimetres, which is 90 centimetres. So that's magnification. Resolution is different, and resolution is really what causes the main downfall of optical microscopy, and why we had to look at other ways of magnifying things. And the resolution is the distance apart that you can see two things as being separate. So, for example, these dots are clearly separated. You can see that they are distinct dots. These dots, yeah, it might be being a bit more difficult to see whether they are two individual dots or one dot. If I do two dots even closer together, that now is looking like it might be one dot. And that's resolution. So you can get a really good idea of resolution by using a digital camera on its lowest resolution setting, because otherwise you'll be there all day magnifying things, or looking for low resolution images on the internet. And if you get a low resolution image and zoom in, it becomes pixelated. You get to the point where you can't see where one thing finishes and the next thing starts. And in microscopy, that limit is based on the wavelength of light. So, light is a wave with a certain wavelength. And in order for us to get a good image of something, we need to be able to see the point at which two things are separated. And in order to do that, you need to be able to get the light to go in between them. So, the resolution of an optical microscope is limited by the wavelength of light, and that is an absolute limitation. No amount of improvements in lens technology or increased magnification is going to get you a resolution that is better than this, and that number is 200 nanometers. So that is the absolute limit of a light microscope. If you want to go further than that, then you have to look at other microscopy techniques, and we're going to talk about those in another video. But the light microscopes still used today because they're cheap, they're easy to handle, they're robust, they don't need an awful lot of sample preparation, so at the very least you can get your sample of onion skin, put it under your microscope and see some cells. It works even better if you stain it, but that is the most you need to do. And as Lau and Hooke did, you can see things moving if they're alive. As I say, I'm going to talk again, do another little tutorial on electron microscopy, but light microscopy is an old technique. I mean, the first publication from it was in 1665, but it's still very relevant. And lots of schools and colleges and all universities, I would say, would have light microscopes that you can use as a student, which that's not the case for electron microscopy. So I hope that has been interesting and beneficial and has given you a bit of a lowdown into the techniques of microscopy. And next time we'll have a bit of a look at electron microscopy.